There are two techniques for measuring hematocrit, and you must practice both. This particular method uses an old-fashioned metric ruler. Essentially, we will make two measurements and perform simple math. I've noted a generic formula for you here. Our first measurement, x, will be divided by the second measurement, y, and the result multiplied by 100. You can measure in either millimeters or centimeters, just be consistent. Our first measurement here is the height of the erythrocytes. No, don't include that little clay plug in this measurement. You're going to measure from bottom of red to top of red. Our example measurement here is 6.4 centimeters. The second measurement is from the bottom of the red to the top of the plasma. Once again, do not include the clay plug. Our example measurement here is 11.7 centimeters. Your last step is to divide the first number, the packed erythrocytes, by the second number, that whole blood. Once you're done with that, multiply by 100 and you have the percent hematocrit. So that would be 6.4 divided by 11.7 times 100 is 54.7% hematocrit. It's important to note that the unit is just as important as our number. Always make sure you write the appropriate unit and show all measurements and calculations in your answers. The second method references our two measurements, the bottom of the red to the top of the red, and the bottom of the red to the top of the plasma. This time, instead of directly measuring those landmarks, we're going to use them in placing our tube on a CritiCaps card. Okay. Take a close look at the CritiCaps card, and you'll notice that each line on the grid corresponds to numbers on the edge. Basically, we're going to place our tube and read a number off this chart. The key to this method is accuracy in the placement of the tube. The bottom of that red should be lined up with the 0 line on our grid. Bottom of red on 0. Slide the tube along the grids until the top of plasma aligns with 100. Bottom of red on 0, top of plasma on the 100. Your next step is to find the line on the grid that runs across the top of the red. You're measuring the top of the red. Follow that line to the edge and read the number off the card. I'll erase everything here, and we can see that that line that goes across the top of the red is 47%. This is 47% hematocrit. Regarding hematocrit, both males and females have normal ranges. The normal range for females is 38 to 46% hematocrit, while males are higher at 42 to 56% hematocrit. A hematocrit lower than the normal range is interpreted as anemia, polycythemia refers to an elevated hematocrit. Let's switch subjects to blood typing. Human erythrocytes can contain self-antigens as cell receptors. These self-antigens define one's blood type. Individuals with self-antigens A are defined as blood type A. Those with self-antigens B are defined as blood type B. Individuals with both self-antigens are blood type AB, and individuals with no self-antigens are blood type O. There's a separate self-antigen gene inherited independently from these. That self-antigen is Rh. Individuals with Rh antigen are noted as a positive, while those without Rh antigen are considered negative. Therefore, an individual blood type is always noted as a letter and a plus or minus sign. The trick becomes, how do we determine which antigens are present? Well, we take advantage of the immune system. When we're exposed to an erythrocyte antigen that does not belong in us, our B lymphocytes make antibodies against it. Those large antibody proteins will bind to the antigen in a process called agglutination. If we put human blood on a plate with pre-made antibodies, we can see that agglutination and use it to see if an antigen is present. This picture shows that blood test. A few drops of the same blood is placed into each of the 3 wells. Into each well, we have added one antibody against that antigen. The clumping you see in each of these wells is a result of agglutination. This tells us that the antigen in that well is present. 2 test samples. To the right is a test on real human blood. Agglutination with real blood is easily seen in each well. This test shows that all 3 self-antigens are present. So this blood type is A, B positive. On the left, we see a test using simulated human blood. To interpret this test, look to see if you can see a change in the well. If that well has changed, that is a positive test result. This type is B negative. Why? Well, the B well has changed, while the R and RH wells have not changed. Interpreting simulated blood is a little more challenging, as the simulated agglutination is more of a texture change than real clumping. That binding of antigen and antibody could occur in our bodies if we are exposed to erythrocyte antigens that are foreign. For example, someone who is blood type A contains self-antigens A. If that person is exposed to blood with a B self-antigen, they'll create antibodies against that blood. Therefore, it's imperative that compatible blood types be used in the clinical setting. Professor Imholtz gets the credit for this diagram, and it does help with determining compatible types for donation. Recall, blood type O occurs when there are no antigens. You can think of O negative as generic blood. Those O negative erythrocytes have no antigens and can therefore be given to everyone else. When looking at either A or B, those can be given to themselves, obviously, but they can also be given to an AB positive. Why? Well, someone who is AB already has the A antigen, so there's nothing foreign. That same AB person already has the B antigen. Again, there's nothing foreign. But that compatibility does not go backwards. For example, you cannot give AB blood to someone who's just an A because the B is foreign. You cannot give AB blood to someone who's just a B because the A is foreign. You cannot give AB blood to someone who's an O because both A and B are foreign. On the upper left corner, we see how the Rh antigen compatibility works. Rh positive individuals can donate to other positives because it matches. Rh positive individuals have the antigen. And so again, they can donate to another positive of a compatible letter type. But someone who is negative does not have the Rh antigen. You cannot give them a blood type that has a positive. Think of Rh this way. You can give a positive blood that is negative, but you cannot give a negative blood type, blood that is positive. So I'll repeat that. You can give positive a negative, but you cannot give a negative positive. On the next slides, we will practice identifying the blood type based on the blood test results. In addition, you should be able to describe each blood type in terms of the antigens present, antibodies made, other types this blood can donate to, as well as types this individual could receive from. For example, A positive blood contains A and Rh antigens. It would make B antibodies if exposed. A positive can donate to A positive because it matches. It can also donate to AB positive because everybody can. A positive can receive A positive because it's compatible, it matches. It can also receive O negative because everybody can. With the Rh rules, A positive can also receive A negative. And A positive can also receive O positive. But A positive cannot receive any blood containing a B antigen. Here we have simulated blood tests. That red blotch indicates agglutination. If you see that blotch in a well, it indicates that the antigen is present. Let's practice some data analysis here. I'll show the diagram, give you a few seconds, and let you know the type. Look at this one. Rh antigen is present, but nothing else. So that is an O positive. Perhaps pause the slide or pause the video here so you can think this one through. On this one, we can see A antigen is present, B antigen is present, and Rh antigen is present. This type is A, B positive. So AB positive is considered the universal recipient. It has all antigens and can receive all blood types. This one is the opposite. If we look at this one, notice how there are no antigens present. There's no agglutination in A, B, or Rh. This blood type is O negative. O negative is considered the universal donor. Because it's kind of generic, it has no antigens, it can be given to all other blood types. So to wrap up, you need to practice at this point. We highly recommend the nobelprize.org typing game for practice. You can choose patients, draw and test their blood, Then choose and hang blood bags by type. I hope this was helpful in this short video. Again, at this point, you need to focus on what's in the lab manual and do a lot of practice. Take care and be well.